Hello, fellow warriors. Here we are with news from the website prostatewarriors.com.
It is great to be here. There's just a massive amount of new data to get through.
There really is. I mean, for decades, the medical world has looked at these, shape-shifting proteins that drive aggressive prostate cancer and basically labeled them as undruggable.
Right, which is a devastating word to hear. But, well, today we're looking at the science proving that word is officially obsolete.
Exactly. So our mission for this deep dive is to unpack this huge wave of research. And we know your time is valuable, so we're starting right on the front lines.
Yeah, we're jumping straight into the latest clinical trials before zooming out to some really wild preclinical breakthroughs.
Things that are literally rewriting the rules of modern oncology. So let's start with those direct human applications, specifically a phase 1 trial for a therapy called BPX-601.
Right, so BPX-601, this is a completely new way to control the immune system. It's a next-generation CRT cell therapy.
And this is specifically for advanced prostate cancer that stopped responding to traditional stuff, right? Like anti-androgens.
Exactly. Now, traditional CAR T-cell therapy involves taking your own immune cells, reprogramming them in a lab to attack cancer, and infusing them back in.
Which sounds great on paper.
It does, and historically, it worked pretty well at finding the tumor. I mean, earlier iterations saw 56% of patients hit a 50% or greater reduction in their PSA.
Wow, that's over half the patients seeing a massive drop.
Right, but the problem was those supercharged cells didn't have an off button, so at higher doses, they often triggered really severe runaway immune complications.
Oh, because they were just aggressively attacking everything. So how does this phase 1 trial fix that?
Well, BPX-601 has this unique built-in safety mechanism. Researchers are calling it a go switch. It lets doctors physically turn the reprogrammed cells on or off using a totally separate drug.
Oh, that's brilliant. It's like having a remote control for a guard dog.
That's a really good way to put it, actually.
Yeah, you want the dog to be fierce against intruders, which is the tumor, obviously.
Yeah.
But you absolutely need to be able to call it off before it, damages the house.
Exactly. This trial is heavily focused on finding the safest dose because now clinicians can actively manage your safety in real time.
That go switch is a huge safety net for live cells. But what if we just want to send like a dumb guided missile straight to the tumor?
Then you'd be talking about antibody drug conjugates, or ADCs.
Right, and there's a phase 2 trial for that right now. It's the ARTEMIS-003 trial.
Yes, and they are evaluating an ADC drug known as HS-200-093.
Let me just pause and spell that out for everyone taking notes. That is HS-200-093.
Perfect. And just so you know, you might also see it called Resvutatug Razatecin, or GSK5764227.
Okay, so how does this guided missile actually work?
It's an incredibly elegant mechanism. It's basically an engineered antibody physically linked to a highly toxic chemical payload. The antibody seeks out a specific protein on the tumor cells called B7H3.
But the tricky part, I imagine, is keeping that toxic payload from breaking off in the bloodstream, right?
Exactly. That's the main engineering hurdle. You don't want to poison healthy tissue. So the linker connecting the toxin is designed to stay completely intact in the neutral pH of your blood.
Oh, so it only releases the bomb when it hits the target.
Right. Once it latches onto the B7H3 receptor and gets pulled inside the acidic environment of the cancer cell, it breaks down. It destroys the cell from the inside out.
And the clinical data from the ASCO GU conference on this was pretty staggering.
It really was. For patients previously treated with taxane-based chemotherapy, the confirmed objective response rate was 38.9%.
And the PSA 50 response rate, meaning their PSA dropped by half or more, was 40.7%. But it gets better if you use it earlier, doesn't it?
It does. In taxane-naive patients, those who hadn't been battered by that specific chemo yet, the objective response rate jumped to 50.0%, and the PSA 50 climbed to 64.0%.
Which is just huge. And because of this, HS-27093 recently got a breakthrough therapy designation in China for metastatic castration-resistant prostate cancer.
Specifically for patients previously treated with endocrine and taxane therapies, yes.
Now, let me push back a bit here. I hear breakthrough therapy thrown around in press releases all the time.
Sure, it sounds like a marketing buzzword.
Exactly. Does it actually make a practical difference in getting this from the lab to the patients who need it? Or is it just a shiny badge?
No, it carries genuine regulatory weight. It's a legal fast track. It essentially means the health authorities recognize this drug addresses a severe unmet need better than existing options.
So it actually cuts through the red tape.
Absolutely.
Yeah.
It obligates the agency to accelerate development and expedite the review process to get it to the clinics faster.
Getting through red tape is exactly what we want. So ADCs deliver a chemical payload, but what about delivering targeted radiation?
That brings us to our next clinical trial, the Phase 2 OPTIMAL-PSMA trial. They're looking at TLX597-TSMA.
Also known as 177-Ludoda Hynek pan-PSMA. Right.
That's the one. It's a next-generation PSMA targeted radioligand. Now, PSMA is a very reliable target on prostate cancer cells. But the real win here isn't the target, it's the dosimetry.
Dosimetry, that's how the radiation is distributed in the body.
Exactly. The data shows this drug achieves high tumor uptake, but delivers a relatively low radiation dose to the salivary glands and the kidneys.
Wait, let me clarify. So the big leap here isn't finding a new target, since it's still aiming at PSMA. It's building a much better delivery vehicle that drastically reduces collateral damage.
You nailed it. Standard radioligands often cause brutal side effects, like severe dry mouth or kidney strain. And those side effects limit how much of the drug a patient can tolerate.
Right, you can only push the body so far.
But by sparing those healthy organs, clinicians can practice dose intensification. They can safely hit the tumor with a much higher, more potent dose of radiation without wrecking the patient's quality of life.
That is incredible. And, it brings up a fascinating point about targets. We just talked about hitting B7H3 and hitting PSMA.
Right.
But why do some of these targets work so well, while others just seem to fail over time?
It's because tumors are highly adaptive. Under the pressure of a drug, they mutate, and the target protein just vanishes or changes shape.
But there's this new observational study in clinical cancer research that explains why B7H3 is becoming, like, the ultimate bullseye.
Yes. B7H3, also called CD276, is incredibly stable. It's expressed consistently across the entire spectrum of the disease, from hormone-sensitive to castration-resistant, neuroendocrine, and even double-negative subtypes.
So it has very low variability. The cancer can't just easily drop it to dodge the treatments.
Exactly. And the researchers proved this using JU Panorama, which is this massive single-cell atlas. They looked at nearly 1 million cells from 213 patients.
Which they've actually made into a free, interactive, R-Shiny web app for researchers globally, which is just so cool. But the mechanistic twist here is how B7H3 reacts to our standard treatments.
Right. So standard androgen deprivation therapy, or ADT, works by starving the tumor of hormones. But the researchers found that ADT actually physically boosts the accessibility of B7H3.
Wait, really? So combining B7H3 inhibition with standard ADT creates a synergistic tumor suppression.
Yes, it works incredibly well together. Plus, because B7H3 is so reliably there, it scores very highly for bispecific antibodies when paired with targets like TROP2, Nectin-1, KLK2, or Nectin-4.
It's like cutting the power to a building to stop the bad guys, which accidentally triggers all their emergency neon vests to light up.
That's a great visual.
It just makes them incredibly easy for our therapies to spot and hit.
You're literally turning the cancer's own survival biology against itself.
Which makes you wonder, if B7H3 lights up when hormone therapies fail, can we just fix the failure of the hormone therapy in the first place?
And that takes us to a new Nature study from UBC and BC cancer researchers. They are going after the androgen receptor itself.
But a specific part of it, right? The part that's always been considered completely undruggable?
Right. So the androgen receptor, or AR, powers 80 to 90% of prostate cancers. Standard drugs hit the rigid ligand binding domain, or LBD.
But the cancer just mutates that rigid shape to resist the drug.
Exactly. So researchers have finally targeted the intrinsically disordered transactivation domain, the NTDT.
And why was that considered undruggable before?
Well, because traditional drugs need a rigid molecular pocket, like a lock for a key. But the NTDT is a shape-shifting floppy protein. It moves around constantly like a wet strand of spaghetti.
So how do you stick a key into a lock that's always changing shape?
You design novel small molecules that bind up to 1 million times tighter than anything before. They have what's called nanomolar potency.
Wow, so it just freezes that floppy domain completely inactive.
Yes, and it vastly outperforms older inhibitors without causing major toxicity.
Wait, hold on. If disordered floppy proteins drive other massive diseases like Alzheimer's and heart disease, does cracking this spaghetti problem in prostate cancer give us a blueprint to fight those diseases too?
Absolutely. It drastically expands the entire druggable universe. It's a monumental proof of concept for structural biology.
That is just mind-blowing. Okay, we have one more preclinical breakthrough to cover, and it's about eradicating resistance
Mm.
by trashing a transcription factor called SOX2.
Right. SOX2 is a huge problem. It drives cancer stem cell maintenance, metastasis, and pushes tumors toward aggressive neuroendocrine phenotypes.
And it helps the cancer evade powerful chemo drugs like docetaxel. But now there's a preclinical drug, GXBP1.
Yes, GXBP1 is a BioPROTAC-based degrader. Instead of just inhibiting SOX2 by blocking it, this drug sends it for ubiquitin proteasome elimination.
So instead of just putting a temporary roadblock in front of SOX2, GXBP1 tags it and throws it straight into the cell's garbage disposal.
Precisely. It completely shreds the protein, and the results are incredible. As a monotherapy, it gave 70% growth inhibition.
And when they combined it with carboplatin and paclitaxel, it achieved up to 96% inhibition.
But the real kicker is when they paired it with osimertinib. It completely eradicated the cancer stem cells and prevented tumor regrowth entirely.
Completely eradicated. I mean, synthesizing all this, we are truly entering an era of unprecedented precision.
We really are. From giving SARA T cells a go switch, to dialing in radiation with TLX597TX, to building chemical staplers and cellular garbage disposals.
We are hitting targets we used to think were utterly invincible. Which leaves you with this final thought to mull over.
What's that?
If we're now able to drug the undruggable and turn a tumor's own survival tactics into a brightly lit target, is the future of oncology less about discovering new weapons and more about tricking the cancer into destroying itself? Stay strong and fight on.